Okay, let's continue with the immune system. This is immune system 2, 5 o'clock. Okay, innate immunity. We're still on innate immunity, non-specific immunity. Now we're up to antimicrobial proteins, so which are interferons. This is on page 867 to the right. Interferons are proteins produced by virus-infected cells. They really act as paracrines when they put out in a neighborhood and they attach to other cells and they warn other cells, essentially, that there's a virus in the neighborhood that I'm infected in, and take care of yourself. And cells can make certain antiviral proteins and protect themselves somewhat against viral infection. So it's a warning. And then they also stimulate NK cells to cause apoptosis in virus-infected cells, and it stimulates macrophages and helping destroy virus-infected cells. So this is something that your immune system can do. It actually destroys your own cells that are infected by virus, so they can't make more viruses to infect other cells. Okay. Complement system. This is a diverse array of proteins. These are inactive proteins that's floating around in your blood, and they're activated two ways. There's an intrinsic, a classical pathway, and an alternative pathway. It's that important to understand that. One is which a complement protein binds to an antibody that's attached to an antigen, and sometimes they can be activated just by the surface proteins of bacteria, can activate complement. And complement then can do several things. One, cytolysis. It can actually insert certain proteins into a bacterial cell wall and cell membrane, and actually make what's called a membrane attack complex. Makes a big hole in the membrane and let everything leak out and attack it, and can lyse foreign cells. It also can stimulate inflammation. Now we're going to be talking about inflammation a bit in a later slide about what inflammation does. Immune clearance helps to attach to antigen antibody complexes and stimulate phagocytosis of those. They can attach to erythrocytes. That's right. The antigen antibody complex attaches to erythrocytes, makes their way to the spleen, and the spleen destroys these. Also, opsonization. This is on page 868. Binding of a protein complement to a portion of bacteria. It covers these complement proteins, cover bacteria, and it stimulates macrophages or other phagocytic cells to engulf this bacteria and destroy it. So stimulating of phagocytosis. All right. Here's a picture of a virus-infected cell that's releasing these different types of interferons to help normal cells synthesize enzymes that interfere with viral replication, to stimulate an NK cell to cause apoptosis of a virus-infected cell, or stimulate a macrophage to phagocytose a virus-infected cell. And here's the, just repeating on the bottom of page 868, the 4 ways that complement helps to protect your body against foreigners. Opsonization, increases inflammation, cytolysis, and elimination of immune complexes. Okay, so let's go more into inflammation. And that also starts on page 868, goes on to 869, and then 870. So first, the cardinal signs, that actually begins at 870 to 871. Redness due to increased blood flow, heat, swelling, pain, and loss of function. Inflammatory and chemotactic factors are certain chemicals that are released by damaged tissues. One of those is called bradykinin and other kinds of kinins that what they do is they increase capillary permeability, so more fluid leaves capillaries. That's one reason why damaged areas swell up. And then chemotaxis, this is cells following a chemical signal. They can detect chemical signals and can find a way to where these chemical signals are concentrated. And so that helps bring more white blood cells to the area by chemotaxis. Mobilization of defenses, hyperemia, which is just bringing more blood to the area. Arterioles open up, capillaries open up, bring more blood to the area. This is one of the reasons why it gets hotter. CAMs, cell adhesion molecules. These are proteins that are made by the endothelial cells lining the capillaries that cause white blood cells to stick to the inside of the capillary. This is called marginalization. CAMs on leukocytes adhere the CAMs on the endothelial cells of capillaries, and they call it cellular Velcro. I'm on page 869. Then these white blood cells are squeezed between the spaces between the endothelial cells. This is called diapedesis. Okay. And then there's delivery of plasma proteins, clotting factors that are brought there to start the clotting cascade. Kinins to increase capillary permeability and cause more fluid to be produced there. This is called exudate, which is extra fluid coming out of capillaries. And then there's tissue cleanup and repair. There's growth factors within platelets. Now, during this time, platelets are going to be sticking to the inside. The blood vessels are going to be degranulated and release these the things we talked about earlier in the blood chapter on platelets, factor 12, thromboxane A2, and a growth factor. Now, growth factor stimulates mitosis. And so we're going to have to stimulate mitosis in the smooth muscle of a damaged blood vessel in the connective tissue that's around the area to repair this area. Here is the figure on page 869. So you can see release of inflammatory and chemotactic factors from mast cells. They will attract more white blood cells to the area. They will cause the capillaries to be much leakier, a lot of fluid coming out. This is part of 2 on that figure. Vascular changes include vasodilation of arterioles, increase in capillary permeability, and display of CAMs, cell adhesion molecules. 3, recruitment of immune cells, marginalization, the sticking of these white blood cells to the inside of the capillaries, diapedesis, then moving out, leaving the cardiovascular system, going out into the tissues, and chemotaxis following these chemical trail to the bacteria that has entered through their injury. Remember, every time you cut yourself, bacteria is going to get into this wound. And delivery of plasma proteins, clotting factors, and kinins and inflammatory chemicals. And what happens is formation of exudate and wash of infected area. So this greater permeability causes a lot more fluid to come out into the tissue. And what this does is it actually washes it. It's like a little current that carries bacteria away from the blood vessels and into a lymphatic capillary. We want to wash these bacteria or viruses or whatever is getting into a lymphatic capillary. So actually, when you get injured, bacteria do not enter your cardiovascular system. And this exudate prevents that. Actually, bacteria or other pathogens in your blood supply is called sepsis. And that is really a very serious condition. Bacteria and viruses and everything are swept into lymphatic capillaries. And that's a good thing because they will be percolating down to the lymphatic system and eventually hit into lymph nodes because the lymph goes to lymph nodes. And lymph nodes are loaded with immune cells, white blood cells, phagocytes, B and T lymphocytes. And these pathogens are going to bump into a white blood cell with the correct receptor, and then we're going to have adaptive immunity or specific immunity, which we're going to get onto in a little bit. Okay, so more on this slide is more on inflammation. Increases in interstitial hydrostatic pressure causes increased uptake of fluid by lymphatic capillaries. Because all this exudate coming out there. All remaining organisms, substances put in the lymphatic system to be dealt with in lymph nodes. And this exudate is akin to washing the air, flushing them into lymphatic capillaries. Okay. Then later on, we'll get into what's happening in the lymph nodes with the B and T lymphocytes adaptive immunity. But first, let's talk about a couple, one more non-specific defense, and that's fever. Okay. There are so-called pyrogens. Pyrogens cause a fever. There's exogenous pyrogens, which are really proteins on the surface of bacteria, and endogenous pyrogens, ones that are made by your own body to induce a fever. And it used to be thought that fever was like a bad thing, that it was something to be fought and reduced. And prostaglandins are one thing that cause fever, and aspirin actually inhibits the production of prostaglandins, so you can actually break a fever by drugs, by one of which is aspirin. But now it's thought that fever is a natural response to infection, that your body temperature is actually, your thermostat is reset to a higher level, and this slightly higher body temperature helps you fight these invaders. So it's a natural body mechanism, and now the thought is you really should let it run its course. If it's a mild fever, sometimes they can get really a high, like you get up to above 100 degrees Fahrenheit, 104 or something. Then something has to be done. It can get out of control and get to be dangerously high. But if it's just a mild fever, the thought is now let it run its course. And there are stages: onset, where your body temperature starts to rise, right? And then stadium, where it's set at that higher level. And you defend that level. That's why people feel a little, who have a fever, feel a little chilly in a room that's other people feel quite comfortable in, because their body temperature has been reset. Their body thermostat is higher. Now that, and that stays to a certain higher to a certain number of days or whatever until you fight off this infection. And defervescence is your body temperature not coming back down and being reset at the normal place at 37 degrees or 98.6%, 98.6 degrees Fahrenheit. The benefit is it inhibits the reproduction of pathogens at this higher temperature, increases the production of CAMs. Okay. But the risks, you can cause metabolic pathway disturbances, denaturization of proteins if it gets too high. So then if it does get too high, you need to attack it and bring it down. Okay. Now we get on to specific defenses, adaptive immunity, general aspects, specificity. Here we are going to attack specific organisms that have specific antigens. Antigens, remember, are foreign proteins. We can recognize specifically as foreign proteins. And then memory is another aspect of it. We are going to be making cells that will remember our exposure to this pathogen. And the next time we're exposed to it, we can attack it right away and not never even realize that we've been exposed to it again. And so we can get sometimes lifelong immunity after being exposed to this particular pathogen, living through it. And then we have maybe antibodies, and we have T cells that remember this, and we never have to worry about it again. That's the hope with coronavirus, that if you do get it and you live through it, you are protected from that organism, that pathogen for the rest of your life, as long as it doesn't mutate. Here's the problem: we have different strains of these organisms, and so that's why every year there's a new flu strain, and you might have become immune to the last year's flu strain, but it's changed the membrane proteins, it's a new antigen, and we got to go through the whole process again. Okay, and so this is called delayed immunity. Remember, before, the innate immunity was right on at the moment. This is delayed immunity because these pathogens have to be swept into lymph nodes, and they have to be get into the bump into the right lymphocyte. This lymphocyte has to be activated. The lymphocyte has to go through mitosis and make many more of itself, and then it can attack this pathogen and take up. So in the meantime, we could be miserable. Really, really feel sick until we can mount that defense, and then we can wipe it out. Okay. Forms of immunity. There's two types of adaptive immunity: cellular or cell-mediated immunity, and that's T lymphocytes. These guys can only destroy cells. That's it. So they destroy bacterial. They can destroy bacterial cells, fungal cells, and cells that have become infected by a virus or become transformed by cancer. Humoral is also called antibody-mediated. The cells that do this are B lymphocytes that make antibodies, that tag antigens and the pathogens they're attached to for destruction. All right, so what are antigens? Antigens generally are foreign proteins. But there's sometimes, but we also have self-antigens. And actually, lots of the proteins inside your cells will be self-antigens because your immune system doesn't recognize those. In the later part of this chapter, we'll learn how when the immune system develops, when you're an embryo and a fetus, it has to go to school to learn what self-proteins are and to not recognize those. All right, so you have what's the word I'm looking for? Okay, I can't come up with that word, but it's inability to recognize self-protein. So you don't attack yourself. Sometimes it makes a mistake and does attack a self-protein, thinks it's an antigen. And that's when we got autoimmune disorders, like type 1 diabetes, like rheumatoid arthritis, like multiple sclerosis. But actually, any cell any protein inside your cells is actually a self-antigen because your immune system is just an extracellular system. It doesn't see the proteins inside your cells. It only sees the proteins on the cell surface. That's it. This is why when a virus gets inside a cell, it's protected from the immune system. That's too late. The immune system cannot attack a virus that has already injected its nucleic acid into a cell. How we can wipe out viruses is that they cause the cell to make new viral particles and they lyse that cell. And then once these viral particles are out into the interstitial fluid, then they can be attacked by antibodies, but not when they're inside cells. And so some viruses incorporate, called constituent viruses, that incorporate the nucleic acid into our chromosomes, and we have them for life. When they become active and make viral particles and they leave the cell, then we get that. Right? Okay, determinant and haptens. You don't have to worry too much about this. Determinant is the part of the protein that your immune system actually recognizes. And there's some small molecules called haptens that are actually smaller than proteins that are antigenic. And so some things that, like I said, most antigens, most things that your immune system attacks are proteins. But there's sometimes other ones, like poison ivy, for example. That's an inflammation due to an oil on the leaves that is antigenic, that we have an immune response to. It's kind of odd, but that's it. So immunogenic response. So an immunogen is something that precipitates an immune response. And these are usually foreign proteins. All right, lymphocytes. So these are cells that conduct adaptive immunity. They have receptor complexes, T cell receptors and B cell receptors. These are receptors for foreign proteins. It is kind of amazing. The immune system is kind of amazing in the sense that you have in your lymph nodes lymphocytes that have receptors for foreign proteins on bacteria and on viruses that you have never encountered. But your ancestors have encountered them, and they have, and if they happen to have a receptor that fits this particular foreign protein in these pathogens that are out there, your ancestors lived through that. And they passed on those genes that make those particular proteins so that we have them and can attack these foreigners. T cells have co-receptors, CDs. These are receptors that bind other cells, and when we get to the T cell receptors, we'll learn that there's two types, CD4 and CD8, killer cells and helper T cells. So toxic T cells, which kill cells, and helper T cells that help other cells, and they have different CD receptors, co-receptors on them. Okay, T cells have to be antigen presented. And I just mentioned, there's two types of T cells, helper T cells and cytotoxic T cells. And they have different co-receptors, one of them CD4, which is helper cells. Sometimes they're called CD4 cells. And then we have the cytotoxic cells that have the CD8 receptor, and sometimes just called CD8 cells. Okay, now this antigen presented, I'm going to get into that in a later slide, and probably in a later video, how in order to activate these lymphocytes, antigen has to be presented. And that is mixing part of the foreign protein with a self protein and putting that into the cell membrane of the APC. APC stands for antigen presenting cells. And one of the APCs are B cells. B cells themselves are antigen presented. And this is a way of activating white blood cells. White blood cells have to be activated, not once, but actually twice. And we're going to get into that later. And we're going to get into the whole antigen presenting process with what are called MHC proteins, major histocompatibility complex proteins. And these are actually cell ID proteins. These cells identify your cells as belong to you and no one else. And everyone has a unique set of these, unless you have an identical twin. And these proteins are essentially why you can't just share organs with anybody, because you can recognize your own MHC proteins as belong to you. This is self-tolerance. That's what I was looking for. We have self-tolerance for our own proteins. We have no tolerance for anybody else's proteins. And that's one, unfortunate part of the immune system. It's dumb in one sense, in the sense that if it's foreign, it's bad, and no questions asked. Doesn't matter whether it's an innocuous foreign protein, as I mentioned before, in allergies, or helpful foreign proteins that might be on the surface of an organ that we'd like to have, but it belongs to somebody else and has different MHC proteins. So you have to find somebody with a very similar set than you, like you. Sometimes you can find within your own family. Other times that doesn't work, and you can find some stranger who actually has a very similar set of MHC proteins just by chance. And you could actually share organs, still taking some immune suppressive drugs for the rest of your life, but you can live with that organ. Okay, this slide shows you some T cell receptors. T cell receptors There's a helper T cell with a T cell receptor for TCR stands for T cell receptor for a specific foreign protein on a specific foreign organism. And then the CD4 protein is the helper, is the co-receptor. And it designates the class CD4 as a helper T cell. And co-receptors, as I mentioned, you have to, in order to activate these cells, you need two steps, two forms of activation. One, to activate them and say, okay, then you gotta say, okay, I'm activated. I think there's a foreigner present, but is there really a foreigner present? We gotta have a second activation just to make sure. Cytotoxic T cells, they have T cell receptors also for a specific foreign protein, but they have CD8 receptors. And when they're activated, they're actually activated by helper T cells. Helper T cells are called helper T cells because they help activate other lymphocytes, both cytotoxic lymphocytes, and they help activate B cells also. And the B cells on the right there, they're the ones that make antibodies, and they have B cell receptors. And they will, for a specific foreign protein, an antigen. And they would take in that antigen present, and if they're activated a second activation, they'll start making antibodies. All right. Okay, I'm going to get into antigen presenting. Therefore, that'll be the first part of the next lecture video. So I'm going to call it here and stop this. And I'll see you later in the next video.